Hello everyone, I'm Andrew Schroeder, and this paper is titled Bidirectional Regulation by BACH2 Coordinates Systemic CXCR5 Negative versus CXCR5 Positive CD4 T cell differentiation. So when a naive CD4 T cell is activated, it differentiates into one of a variety of subsets, for example TFH cells, which express CXCR5 and are important for antibody responses, or a subset of non-TFH cells, which mediate cellular responses. And this is an important divergence, TFH versus non-TFH. But what is the mechanism of this divergence? The cytokine IL-2 signals through its receptor CD25 to activate STAT5, which is important for the proliferation and survival of early activated T cells, but it's also been shown to inhibit the differentiation of TFH. And prior studies showed a dichotomy of cells that express CD25 and CXCR5, making the argument that CXCR5 positive cells do not express CD25, thereby avoiding the STAT5 signaling that would inhibit their differentiation. So the current understanding is that this dichotomy is the mechanism for the divergence of cells that will become TFH and non-TFH. However, the first important point of our study is that STAT5 signaling is important for robust non-TFH and TFH responses, and using a STAT5 knockout mouse here, we showed that there was reduced proliferation of cells in the absence of STAT5, and at a later time point there were reduced numbers of non-TFH and TFH cells in the absence of STAT5. So STAT5 is important for a robust TFH cell response. But how do CD25 CXCR5 positive cells develop into TFH if STAT5 can also inhibit their differentiation? Our hypothesis is that STAT5 signals are being filtered and that the factors downstream of STAT5 that will inhibit TFH are being blocked. But what is doing this blocking? We found that the transcription factor BACH2 was transiently upregulated at the protein level after cell activation. And in the absence of BACH2, BLIMP1 was induced at an earlier time point and at much higher levels, both in vitro and in vivo. As BLIMP1 is a known inhibitor of TFH, we would expect to see less TFH in the BACH2 knockout cells. And indeed, we found that there were reduced numbers of CXCR5 positive TFH cells in the BACH2 knockout. We then confirmed that this was due to BLIMP1 by using the BACH2 BLIMP1 double knockout cells, which rescue the differentiation of TFH. So going back, we found that BACH2 was responsible for blocking the signals downstream of STAT5 that would inhibit TFH. So the CXCR5 positive cells avoiding STAT5 signaling was not the mechanism for the divergence. And if we look at an earlier time point, almost all the activated cells were CD25 positive, and the earliest CXCR5 positive cells were CD25 positive, which subsequently downregulated CD25 to yield the divergence that we see. So moving on to the second point, our lab previously showed that BACH2 knockout cells had increased TFH in an intraperitoneal model system. However, I just showed you that BACH2 knockout cells had decreased TFH in an intranasal immunization system. Interestingly, we could find the same phenomenon of promoting and suppressing TFH in the same mouse at the same time. When we looked at draining mediastinal lymph nodes in the intranasal system, we found that BACH2 promoted TFH, that is, the knockout had less. However, when we looked at peripheral non-draining lymph nodes, the BACH2 knockout cells suppressed TFH, the knockout had more. We hypothesized that this was due to lower amounts of antigen and therefore lower IL-2 and CD25 in the non-draining lymph nodes. So to test this, we focused on the same draining lymph node and used a high and a low dose of antigen. And indeed, we found decreased amounts of CD25 and IL-2 in the low antigen dose. And looking at TFH differentiation, in the high dose setting, BACH2 promoted TFH, and in the low dose setting, BACH2 inhibited TFH. So what's going on here? How could BACH2 have this bidirectional function? In a prior publication, we found that BACH2 directly inhibited CXCR5 and inhibited TFH. In this study, we found that BACH2 inhibited CD25 and BLIMP1, which inhibits CXCR5 and TFH. So this is an incoherent feed-forward loop where BACH2 is inhibiting something, but at the same time inhibiting the inhibitors of that thing, thereby indirectly promoting it. So what does this really mean for the immune system? In a strong response with lots of antigen and the draining lymph node, BACH2 allows CXCR5 positive cells to proliferate to STAT5 signaling without being inhibited by BLIMP1, thus generating a robust humoral immune response. Without BACH2, the system doesn't work, and BLIMP1 inhibits TFH. However, in a weak response, where very little IL-2 and CD25 are produced, this pathway is not engaged, and BACH2 directly inhibits TFH. That is, without BACH2, there's more TFH. I think this is the immune system's way of focusing TFH in humoral responses to the draining lymph nodes where they'll be beneficial to the organism, and simultaneously inhibiting inappropriate TFH responses in settings like peripheral lymph nodes, weak infections, or weakly stimulating antigens. So I'd like to acknowledge everyone that contributed to this study. I hope you found this presentation interesting. If so, I'd invite you to take a look at the full article being published in the Journal of Experimental Medicine. Thank you.